right. So, start looking at this and what this is all about. It's important to understand. So we've talked about DNA. We've talked about all the parts of DNA and how DNA gives you your traits and the nitrogen bases, the order of those bases makes you who you are. And then we went into, all right, well, now let's look at how DNA replicates. Okay, and we talked about DNA replication and the different steps to that. Okay, and then even during that we mentioned okay, so why is it important that DNA replicates? And we said, okay, so when your cells make more cells, they need copies of DNA, right? So that's why DNA has to replicate. So today in the discussion, we're going to look at how and why our cells make more cells. So the whole process called cell division or the cell cycle, we're going to look at today and get a better understanding of. Okay. So before we dive into all the terminology and the stages and everything involved, it's good to understand kind of why our cells divide in the first place. So do you guys, when you think about an organism, we're made up of like trillions of tiny little cells, right? Well, why wouldn't we be made up of, I don't know, a million really big cells?
Because then we would be fat.
Well, we would still be the same size. We would just have less cells. Like, why is it good that we have more smaller cells instead of fewer large cells? Ben?
So the cells can replicate.
So that cell replication can happen efficiently. All right. Haley first, then we'll go Luke.
Say if you have a million, when the cells, if they die, then you wouldn't.
It's not affecting as big a part of the organism? Yeah, for sure. So if a cell gets affected by disease or injury, then you're not killing off half the organism. It's just maybe a small portion. Did you have something else, Luke?
That's pretty much what I was saying.
Yeah.
The smaller the cells are, the less damage.
Well, then think about, go ahead, Faye.
Okay, if we didn't have big cells, why wouldn't we be fat?
Well, because, again, think about it. Four markers together make that big of one marker, or you could just have one marker that big. Trillions of cells that make us as big as we are, or bigger cells that would still make us the same size. We just would have less of them. Yeah. So the other reason when we think about cell size and what that has to do with when and how cells divide is this idea of efficiency of the cell. So I don't know, you might not be able to see all of this, but basically if there's a cell that's this big and there's the mitochondria, and then there's a cell this big and there's the mitochondria, and we need to get oxygen to the mitochondria for cellular respiration to occur, okay? Which one's going to get oxygen faster, the smaller cell or the bigger cell? The smaller cell, because the oxygen has less room to travel. So the smaller a cell is, the more efficient that cell will be, okay? Especially at moving materials in and out of the cell. So that's a big reason why evolution has kind of taken care of us to have all these trillions of tiny cells so that they stay efficient and can do their cellular processes in a manner to stay alive, for lack of better words, okay? So that's one of the big reasons there. Another issue with our cells, and if they even will divide, is the type of cell it is. So when you look at let's say, our neurons, our brain cells, right? Our nerve cord cells, they don't replicate. What you have is what you have. Our nerve cells don't replicate. They don't go through cell division because of the type of cell they are. So if there's a spinal cord injury, that's why that's so traumatic, right? And catastrophic. It usually stays that way because the person cannot regrow or replicate that type of cell. But if you cut yourself really bad, that will heal because our skin cells, they're a different type of cell, they do go through cell replication. So a lot of times it depends on the type of cell it is, whether it's even going to do any of these processes. So that's something to keep in mind for sure. Yes?
I have, like, 2 questions. So, can a cell divide just because people are growing?
Absolutely. And you're about to see that. Hang on, I'm getting to that. Oh, second question. Go
Yeah.
ahead.
So then, about the brain cells, our brain grows though.
You get to a certain point. I think they say, I know for males, like it's about 24 that your brain stops developing. So yes, it will, but eventually it gets to a point where there's no more cells made. Yeah. Billions of years of evolution. I don't know. I'm not a medical doctor, so I'm not sure exactly. She asked why don't our nerve cells replicate? They're just not made that way. They're in what's called G0, like it's just a stage of just doing their job. They're not actually in the cell cycle where they're growing and replicating like some of our other cells will. So another thing that could speed up or affect like cell division is injury. Oh, the phone's ringing. Yes, Mr. Crouch. Yep.
Sure.
All right, send her down. Bye-bye. Faith, you're out of here. So if injury occurs, then the cells at that site are going to start dividing even more rapidly than normal because they want to try and heal that site. Okay, so that's another thing that can affect how fast all these processes happen. And then, like Ryan said, growth. From you going from a time as big as you are now, it's not because your cells grew, it's because your cells divided. It's because you have more cells now than you did when you were born. Because your cell division has made you grow. Okay? So that's kind of the why behind why this is important. All of these things here, okay? Another thing that we need to talk about during this unit, and we're going to look at tomorrow a little bit closer when I introduce the project you're going to work on next week, is cancer. The cell cycle and all of these stages that we're going to look at, if they go unchecked and they happen out of control, the result is cancer. Cancer cells form and tumors form. So that's definitely going to be something that's going to be on the test, and we're going to talk about again in the next few weeks before the test, is the idea that if the cell cycle is unregulated and it just happens over and over and over again out of control, cancer is the result. And one of the reasons why cancer is so difficult for doctors to battle is because it's your own cells. Your own cells are the problem. And it's hard to kind of kill off your own cells without killing off a bunch of healthy tissue in the process. Okay? And so I think you're going to do a little research project where it'll be interesting to see, and I'm excited for you guys to present and show what you learn about these different cancers and the treatment that is kind of on the cutting edge. So, but anyway, as far as for our knowledge and the test coming up, make sure you understand. You see any kind of question talking about uncontrolled cell growth or cells growing out of control or lost the ability to control their growth rate. All of those phrases should lead you to the path of looking for an answer that involves either tumors or cancer. Okay, because that's the result. If we don't have the cell cycle under control, the cells just keep replicating and replicating and replicating and making more and more and more and more and more cells when we don't need all those cells. Okay? So a few definitions.
Wait.
Yeah.
So, what happens if you don't treat cancer? Like, how will you die?
Well, it gets the tumors get in the way of internal organs that have functions.
Oh.
Like if you have a big mass in your lungs, it's not supposed to be there. It makes it really difficult to breathe. And so, yeah. That's a good question. A lot of people have talked about that today. So, few definitions. You got cell division, mitosis, and then chromosomes and chromatin. These two I'm going to talk about real quick. So chromosomes and chromatin are synonymous. They're both just different forms of our DNA. So when your cell's just hanging out doing its job, the DNA is in a form called chromatin. Okay? It's really thread-like. It just kind of looks like a mass of threads all bundled together. When your cells start going through mitosis, getting ready to divide into two new cells, that chromatin, the DNA, kind of thread DNA, condenses into more distinct structures called chromosomes. Okay? And so when you hear these two words, DNA, they're just talking about DNA, they're just talking about different forms of DNA. So make sure we realize that, okay? Then cell division and mitosis. Cell division is pretty self-explanatory. It's the cell going from one cell dividing into two identical cells. Now, when we're talking about this process, mitosis and cell division, we're talking about our body cells. Okay, our body cells, what's called our somatic cells. All of your body cells, your skin cells, your bone cells, your brain cells, your liver cells, your heart cells, those are all your body cells, or again, the fancy sciencey word for that is somatic cells. The other type of cell, which is our sex cells, or our gametes, okay, those go through a different process to replicate. But we'll talk about that before Christmas. So, cell division, how the cell divides. Mitosis is basically how the nucleus divides. Okay? How the nucleus divides. Alright? So, let's start looking a little bit more. This is just a quick thing on chromosomes. Every organism has kind of a set amount of chromosomes. And again, this would be kind of your typical chromosome. You got chromatid, which are like the legs and arms, kind of the little pieces we would call chromatid. The centromere is the middle part there where it all meets together. So now we're going to get in kind of to the meat and potatoes of the notes and of the lecture and kind of what you need to know. So there's 3 stages to the cell cycle. And think of the cell cycle kind of like, like a life cycle, right? You're born, and then you're an infant, and then you're a child, a teenager, and you have all these different stages of your life. Well, cells, okay, have the same thing. They have kind of 3 main stages. They have interphase, which has 3 parts to it, mitosis, which has 4, and then cytokinesis, which is just kind of the final thing, okay? So let's start going through what's happening during all these different stages, all right? So the first one, okay, gap 1. This is the first stage of interphase, okay? Gap 1, the first stage of interphase. Basically, the cell is growing. The cell gets a little bit bigger, okay? That's pretty much it. The next stage of interphase is something called the S phase, okay? The S phase. S stands for synthesis. Remember how we talked about DNA replication last week, right? Ta-da! This is when it happens. During the S phase of interphase. The DNA replicates. You go from 1 strand of DNA to 2 strands of DNA, right? Okay? That, I promise you're going to need to know this one. Okay? You might put a little star by the S phase. Okay? Because that is definitely something that you're going to see again throughout the unit. DNA is replicated during the S phase of interphase. Okay? And then the last stage of interphase is something called gap 2 or G2. What happens there is the organelles, all the cell parts, those replicate. Okay? So, but before we move into the next part, I want to make sure you write something down. Something that's not on your notes, but I need you to add, is during gap 1 and during gap 2, there are checkpoints. And checkpoints basically are, think about like you go to a checkup at the doctor and they're checking you up and just making sure you're healthy, right? That's what these checkpoints do for the cell. Okay? So the checkpoints check to make sure that the cell is on the right track to divide properly. And if it's not, then the cell gets killed off by an enzyme. Okay? So if there's some sort of mutation or something not going right, it doesn't get to go past this interphase stage. It doesn't get to enter into mitosis. Okay, so most of the time when these checkpoints are working the right way, this is how we keep ourselves from becoming cancerous. But if a checkpoint is missed or screws up, it can lead down that path to the cell uncontrollably dividing and causing tumors and cancer. Okay, so these checkpoints during G1 and G2 keep the cell from becoming cancerous if there's something wrong with it. Okay, so something about checkpoints happening at G1 and G2. All right, next stage, or the next big part is mitosis. Okay, so mitosis is how the nucleus kind of copies itself and gets ready for the cell to divide all together. Okay? There's 4 stages: prophase, metaphase, anaphase, and telophase. So the first stage, prophase. Remember back a long time ago we talked about centrioles. Okay? Centrioles they look like little churros or like little gears in the cell, y'all said. And I said, oh, we'll talk about those when we get to cell division. Well, here we are. So the centrioles, the first thing that happens during prophase, the centrioles move away from each other. They're going to help the chromosomes kind of line up in the right spot. That's what the centrioles are all about. So they move away to opposite ends of the cell. That's first thing of prophase. The second part of prophase is the nucleus disappears. Okay, so the nucleus disappears. The nuclear envelope goes away. So that's the second part of prophase. Okay? The third part of prophase is that chromatin, that thread-like chromatin, condenses into chromosomes. So that's the third part of prophase. We're still in prophase. Prophase is the longest stage of mitosis. It's a really long stage. And we're still in it. So chromatin condenses into chromosomes. All right? And then the last part of prophase is, remember I said those centrioles are going to help the chromosomes kind of line up? Well, they shoot these little web-looking things. These spindle fibers are created, okay? And then the chromosomes attach so that they can kind of move around the cell to the right spot, okay? And again, you might be like, what the heck is all this even about? It's about getting the right amount of DNA into the 2 new cells that we're trying to create. That's why we go through all these stages. To get the DNA, which remember is the chromosomes, to get the right amount of DNA and the right copy of DNA into the 2 new cells. Okay? All right. Metaphase. Now it's all downhill. Now it's super easy and super short. Metaphase, right down middle. So next to metaphase somewhere, right down middle. Metaphase, middle. That's all that happens. Chromosomes on those spindle fibers move to the middle of the cell. Notice your notes use some different words. Use some synonyms for middle, like center or equator. That all means the middle. Metaphase, middle. Metaphase, middle. Chromosomes line up in the middle. Next part, anaphase. Anaphase, away. All right, so now, look, these chromosomes that were attached together, now they're going to move away from each other. All right? And let me show you something. Like, if you look here, so these were, I'll just use this, so these were just together, right? And we had that centromere, all right? Well, look, this is a copy of that. Remember how in S phase we replicated DNA? Ta-da! That's a replicated piece of DNA. That's its exact copy right there. So as they go away from each other, again, we're setting the stage so that each new cell formed has a perfect copy of DNA inside the new nucleus. Okay, that's what it's all about. That's why we do all these stages. So we get the right DNA in the right spot. All right, and then the last stage is telophase. Okay, and in telophase, basically everything that was undone in prophase is kind of redone in telophase. So remember in prophase, the chromatin condensed to chromosomes. Well, now it's the opposite. In telophase, the chromosomes go back to the thread-like chromatin. Yes?
So our cells could be doing that like right now?
Right now. Absolutely. Some of your cells are doing that right now. Still in telophase. Remember how the nuclear envelope disappeared in prophase? Well, now it reappears.
Whoa.
Okay? And now you have your two new nuclei right there and right there. And look at the cell. See how the cell's kind of starting to pinch in and getting ready to split? Okay? That pinching in there for the animal cell, we call that a cleavage furrow. A cleavage furrow. It pinches in and then eventually it's going to break apart. Okay? So you might label that or write that wherever you drew that picture. That's called a cleavage furrow. That's only for animal cells. Okay?
How does it work in plant cells?
Great question. For plant cells, it's something called a cell plate forms. So because of the cell wall in a plant cell, they are divided by what's called a cell plate, not a cleavage furrow. Okay, not the little furrow. So then the last stage of the cell cycle of the whole thing is called cytokinesis. And all that happens there, bam, it's split into two identical cells.
Wow.
So the cytoplasm finally kind of splits apart, and you get your two identical daughter cells. All right? And that is all there is to it. I know it was a lot. Good job sticking with it. Let me explain what we're going to do.